Welcome back, ladies and gentlemen. So today let's just kind of put down on paper some of the big picture pieces of mitosis and how that compares to meiosis. So when we look at mitosis, remember this is a single round of cell division. So the real function here for mitosis is to conserve that set of chromosomes. We want to keep the chromosome number the same. So let's just use human cells as an example. We're going from 46 chromosomes in the parent cell to two genetically identical daughter cells, each with 46 chromosomes in their cells as well. They're clones. They are clones to each other, they are clones back to that parent cell. And that purpose of mitosis in keeping that chromosome number really depends on the context. So why are cells going through that division? Well, it may be for reproduction purposes, depending on their life cycle and their life history. It could be for repair, it could be for growth, it could be for replacement. Um, so you really have to pay attention to the context in which that organism is going through mitosis to understand the reason why it is making more cells and needing to keep that chromosome number consistent. We go through meiosis, however, and this is pretty much standard reproduction. That's its purpose. And so in order to make this work appropriately, we go through two rounds of cell division because in this case, the organisms who do meiosis are trying to reduce the number of chromosomes. So we're going to take a diploid cell, run it through meiosis because the idea here is to take that diploid number, reduce it down to haploid into those gametes, because the whole end goal is to get those gametes to recombine sexually to produce the offspring. It's really about genetic recombination. And keep in mind that that genetic recombination doesn't just happen when we go to make the offspring by bringing together cells from two different parents, but the gametes that are actually produced within a single parent are also genetically recombined because each of the daughter cells that that parent cell creates via meiosis is going to be genetically different because of things like crossing over and independent assortment that are happening as a result of that meiotic division. So if we look at this, this is just kind of nice big overview picture, right? Mitosis, we're going to duplicate those chromosomes during interphase. We'll line all of those individual chromosomes up. In metaphase, those sister chromatids will split as we go into anaphase, telophase, cytokinesis, and get us daughter cells here at the end. And if we look back here at that original parent cell, they are genetically identical to that parent. They are also genetically identical to each other. I look over here at meiosis, and instead, when those cells duplicate and we get into prophase 1, they'll now synapse together into these homologous pairs, what we call tetrads. And then when we get to metaphase 1, those tetrads are what lines up in the center. Here's where we get the crossing over here in prophase 1. Metaphase 1 takes care of that independent assortment idea because it can be completely independent as to which side the major paternal chromosome goes versus the major maternal chromosome from one pair to the next. We get into anaphase 1 and telophase 1, and we are now down to diploid cells, right, where that N comes in. They are already genetically different from each other compared to that original parent. And then I get all the way down through meiosis 2, separate out those sister chromatids, and now I end up with 4 daughter cells, none of which are identical to each other, and certainly not identical to the original parent cell either. So when we look at sort of unique events to meiosis, that tetrad formation during prophase 1 is a big one. That's going to allow that crossing over to happen and allow us to initially start mixing up genetic information between the chromosomes that your parents gave you. So between chromosome 1 and chromosome 1 from your mom and your dad, we're going to start mixing up some information there. Between chromosome 2 from your mom and chromosome 2 from your dad, we're going to start mixing up some information there. And that's what that crossing over allows us to do. We create what they call recombinant chromosomes as a result of crossing over. And then we get to metaphase 1, And now those tetrads, those homologous pairs, are what lines up at the metaphase plate, and it's the homologous pairs that separate. And this is the sort of idea here with the independent assortment, because the way in which your paternal and maternal chromosomes line up in those homologous pairs, in terms of sort of saying this one goes off to the left, this one goes off to the right, they are completely independent. So it's not predetermined that all of your maternal chromosomes have to go to the left and all of your dads have to go to the right. It's completely random. And it's completely random from one round of meiosis to the next as well. And the same thing can be said with the crossing over. How much crossing over happens between those homologous chromosomes, where that crossing over happens, is all random. Keep in mind that those sister chromatids don't end up separating until anaphase 2, um, but that will then ultimately result in those recombinant cells at the end. So a big contributing difference between mitosis and meiosis is this formation of a tetrad. So when we talk about tetrads, right, they are your homologous pairs that now come together. So this entire structure is your tetrad. So you have your original chromosome from your mother, that duplicates during interphase, and so now you have two sister chromatids that are maternal. And then you have the original chromatid from your father, the original chromosome from your dad, and then that will also duplicate during interphase and come together to make a chromosome that is paternal. When we go through mitosis, this still happens, but these two chromosomes would have stayed completely independent of each other, lined up at the metaphase plate, these sister chromatids would have split, and then those two daughter cells would have each gotten a green and an orange. Difference here is with meiosis, we're trying to half the chromosome number. We're actually actively getting these homologous pairs out of the same cell. We want them in separate cells. So in order for this to work, these homologous pairs have to synapse together. They have to come together and create this tetrad. And this is going to happen during prophase 1. The other major component here isn't just about getting these two line up so that they're connected and we split them during metaphase 1 and we get the homologous pairs in separate cells. That's only half the story. The other component here is that by bringing these homologous pairs together into this tetrad, it is going to much more easily facilitate a more accurate crossing over event. And we'll talk a little bit more about that crossing over in the next video. But the idea here is that we're going to mix up genetic information. So some of this green will end up with some of that orange. We'll create these recombinant chromosomes so that the information that your parents gave you gets shuffled around before you then pass it on to your offspring. So how do we even know that this is possible? So this experiment gets run through in your text, spend some time with it, but basically we're looking at spore formation here. And there were experiments done on how do these things stay together. And what they found is that these cohesins, are these protein complexes, are responsible literally for cohesion, literally for these guys to stick together. So when we look at the differences between mitosis and meiosis, some of the biggest components here are when those cohesins get split. When do we break them down or cleave them? When we look at this in mitosis, those cohesins get split at the end of metaphase, and that is going to allow them to then be separated into sister chromatids and then ultimately into the two daughter cells. When we look at meiosis instead, the chromosome arms in anaphase 1 stay together as those homologous pairs, or I should say the sister chromatids in that homologous chromosome. The homologous chromosomes separate, but the sister chromatids stay together. The centromeres in the center of those sister chromatids don't actually separate until anaphase 2. So being able to label and sort of watch these proteins, adjust what these proteins do, we could see where those things went, and they were able to trace where, in fact, those molecules came apart and when.